Alright kids, so I'm not there today. So we're going to look at simple inheritance while I'm at home looking after kids. Yes, sweet as. Let's go. Okay, this diagram here, we're going to look at this a lot. It's in your textbooks. You can go back to it anytime you want. You don't need to draw it. So we have first cross, second cross. A cross is quite simply when we get one parent and we breed them with another parent. So you guys are the first cross of your parents. Now, Mendel, the crazy Austrian monk, he was talking about peas a lot, so we're going to stick with his pea examples. Okay, so as you know by now, each parent supplies half of the offspring's genes on the chromosomes. Cool. The genes come with different alleles or indifferent alleles. So the alleles we're going to talk about are for the gene. So the gene is the set, alleles are the different versions. The alleles we're talking about are for the genes of color of peas. So in fact, it's the pea pod. So we're talking about green and yellow. Now green is dominant, and it has the, where's my highlighter? Booyah. It's got the capital G allele. Yellow is recessive, which is why it's lowercase, and it's a G. Straightforward. Now, while we're at it, the letter G is entirely arbitrary. We could have gone with Y, we could have gone with F, but the G for green makes the most sense. Okay, so the genotype. This is the combination of alleles you have. Phenotype. This is the physical expression of those alleles. Alright, straightforward. Now, we can have a homozygous dominant, which is GG. This is green. The phenotype of it is green. We can have heterozygous, which is the mix between the two. Hetero means mixed. Homo means same, which is big G, little g. The phenotype there is still green. Okay? Homozygous recessive, that's how we get the phenotype for yellow. So let's have a look. Here we've got 4 offspring. We've got big G, little g. And that has to be big G, little g, because our first cross, we use true breeding. Remember that term? True breeding. And we breed them together, and this means we always get heterozygous. Now, when we cross the 2 heterozygous ones, that's when it gets interesting. You can see we've got the green one down there. But these guys are also green. And that's because they are dominant and recessive. They are heterozygous. Just like their parents were. But then, 1 out of 4 times, which we'll see how it works out next, you get the homozygous recessive. Okay? Alright, so the way we make these predictions are Punnett squares. Now, it seems really simple, but it's not so, it's pretty straightforward. But they can get more and more complicated when we start crossing multiple genes at once. So color of flower, height of the plant, all that sort of stuff. So your parent 1 goes along the top. It's usually where we put the female. Parent 2 goes down the side. It's usually where we put the dad. It doesn't matter for you guys at all. And this is our first cross. So we could have predicted, because we knew that they were true breeding, that everyone was going to be heterozygous. We would have known this. Mendel wouldn't. He had to work that out. But that's what he did. And that's why he was so cool. So the second cross, we're basically mating the brothers and sisters, which is really common in the animal world and the plant world. We've got heterozygous, straightforward. Again, probably the mum, probably the dad. And what we see is our phenotype, we've got 3 to 1, okay? But our genotype, we've got 1, so 1 homozygous, to 2 heterozygous, to 1 homozygous recessive. And that can be, we'll put a little D there so you know what I'm talking about. Alright, that's it. I'm sorry, that should have been using a regular pen, but that's what we've got. That's how you use a Punnett square. And if you're watching this in class, I now want you to move to the questions on page... Should have had this lined up beforehand. That's how you know I'm a quality teacher. Questions on page 87 and 88. Chew through those up to question 19 if you're watching this in class.